Next, we have Lizzie from Warwick. I believe she is here. I'm going to start the introduction, and Lizzie, if you can start sharing your screen.
Yeah.
So Lizzie has studied chemistry at King's College London and is now approaching the end of her graduate studies, doing a joint PhD between the University of Warwick and Diamond Light Source, specifically Beamline 14 with Paul Quinn. She's involved in mapping the interactions of new anticancer drugs and their intracellular targets, and aims to understand their modes of action and potential applicability. She has extensive synchrotron experience and has worked on different imaging platforms. So she will talk to us today about cryo-XRT of metallodrugs in prostate cancer cells. So Lizzie, when you're ready, please share your screen and let's start.
Can you see my screen?
Yeah.
Hi everyone, thank you for the introduction. I'm going to be talking about two of the projects I was involved in at B24. In particular, we're investigating the mechanisms of action and also the mechanisms of cell death of potent metallodrugs in prostate cancer cells. So just a quick recap on why we wanted to use B24 in the first place. At B24, you can obviously analyze your cells cryogenically, so you don't need to use any chemical fixatives or sectioning, so as close to their native state as possible. And remarkably, we can do this down to a 30 to 40 nanometer resolution, so we can actually distinguish between different organelles at a subcellular level. So this was really important for us because we haven't investigated to such a resolution the effect of our compounds on the ultrastructure of these compounds. Importantly, this provides 3D ultrastructural information on cancer cells. So this is both qualitative and quantitative. As has been mentioned before, one of the great things about B24 is you can perform correlative structured illumination microscopy, so super resolution fluorescence, and overlay this with your 3D tomograms. So you can essentially map fluorescent compounds and other fluorophores onto your tomograms. So what we wanted to use B24 for was to investigate the mechanisms of action of, in particular, photoactivatable metallodrugs in prostate cancer cells. So just a quick overview of phototherapy. So essentially, you're using a drug, so a chemical species, in combination with light to treat cancer. Now, phototherapy can be categorized into two groups. So the first one being photodynamic therapy. So in this case, you've got a chemical species or a drug in the presence of oxygen. And when you activate it with blue light, it can generate toxic singlet oxygen species in the cancer cells, eventually leading to cell death. And the second form of phototherapy I'm going to be talking about is photoactivatable chemotherapy. So in this case, you still have a drug which is activated by light, but in this case, your drug is essentially a prodrug, which becomes the active species, usually by the release of cytotoxic ligands inside the cancer cells. So these are the two complexes we were interested in our research group, investigating what they're doing in PC3 prostate cancer cells. So on the left, we've got a cyclomethylated iridium-3 photosensitizer for photodynamic therapy, and also a diazido platinum-4 prodrug for photoactivatable chemotherapy. So just a quick overview of iridium photosensitizers. So I've mentioned you have a chemical species in the presence of oxygen, and you can essentially generate toxic singlet oxygen species locally within the cancer cells. So we were interested in this complex as it has good selectivity for cancer cells over healthy cells, and it has also shown to disrupt levels of glycolytic enzymes inside cancer cells. So what we don't know is the effect of these compounds on the organelles inside cancer cells. So this is what we needed B24 for. So we essentially wanted to investigate any morphological changes in cells treated with this compound under dark, where presumably it's non-toxic to the cells, and also irradiated conditions where you have your cytotoxic effect. So just a quick overview on the sample preparation in our case. So first we had to glow discharge our grids. So these are carbon gold quantifoil grids, which we then would incubate in fetal calf serum, which will essentially promote the growth of the cells onto the grids. So this is typically incubated for 12 hours. Then you can seed your cells onto the grid, so as a monolayer growth onto the surface. Once you've achieved a good confluency, you can then treat your cells. So in this case, we would treat them and then expose them to blue light. Then you would add your trackers so that you can later track them or identify cells by fluorescence microscopy, and also potentially do cryo-sim as well. Then you would blot them with fiducials. So fiducials, for those of you who don't know, are gold nanoparticles of a fixed diameter. So in this case, the diameter was 250 nanometers. And what you do, you'd blot them onto the grids just before you plunge freeze them in liquid ethane, so that you can later use them as a reference when you're performing tomography. So the general workflow for our experiment, so we would first use fluorescence microscopy to identify suitable cell candidates for X-ray tomography. So this would be coupled to a cryo stage, so everything obviously has to be done cryogenically. Then we would load them into the beamline, and we would perform a 2D initial inspection on the grid to work out which cells we want to analyze. So you do this by exposing cells to short bursts of X-rays, so 0.5 seconds, and you generate a mosaic image. From this, you can identify the cells you want to analyze, and also the section of the cell you want to analyze by tomography. So this section is 16 by 16 micrometers squared. And from this, you essentially tilt your sample at different tilt angles. So typically 0.2 to 0.5 degree rotations. And you would basically acquire images of each tilt angle to generate your tomogram. And if you wanted to obviously take this further, you can then perform volume segmentation of your tomogram, and then you can visualize your sample in 3D. So I included this slide because I just wanted to emphasize the amount of detail you can get from tomography. So here you can see a section of a prostate cancer cell where I've shown the mitochondria circled in yellow. So you can actually see the remarkable detail of the cristae in the mitochondrial matrix, which we were quite excited about, I have to admit, when we first saw this, because we hadn't seen this in cryogenic cells before. But what we really wanted to know was the effect of our compounds on the cells. Like, was it changing or altering any of the morphology of organelles inside the cells? And what we noticed initially from the tomograms is that in cells treated with iridium, upon photoactivation, you could see that the size of the mitochondria looks significantly smaller. So to analyze this further, what we then wanted to do was analyze it in 3D. So I did this using a software called Servos, which was, I think, developed at Diamond. So here you can see A and B are the two untreated control cells, where we've got the nucleus in green and the mitochondria in orange. And at the bottom, C is a cell treated with our iridium complex under dark conditions. And then D, you can see the mitochondria are significantly smaller when you've treated it with the iridium drug upon blue light activation. So I have a short video, it's not very long, just to visualize that in 3D, where you're essentially going through the whole 3D volume. Again, you can see the mitochondria in orange and the nucleus in green. So just to summarize the conclusions we made from this data, we could actually get quantitative data on the 3D volume of these mitochondria in these cells. So we could see that they were statistically smaller in the cells treated with iridium upon blue light activation. So if anyone's interested in this work, we have also recently published this. And the second project I was involved in at B24 was investigating photoactivatable anti-cancer complexes. So in this case, you essentially have a prodrug, which you irradiate with light to form the active species. So in this case, we have a platinum 4 complex, which is photo-reduced inside the cancer cells to form a toxic platinum 2 species and also to release toxic radicals. So this is just an overview of what we wanted to use B24 beamline for. So this complex is actually blue fluorescent. So the idea was we wanted to map the fluorescence of this complex. So initially using fluorescence microscopy to select our cells, then use super resolution fluorescence, and then hopefully overlay that with our 3D tomographic information. So again, just an overview. This is normally before your beam time. You perform general fluorescence microscopy just to locate suitable cells on the grid. So here's some examples of 4 PC3 cells which have been cryopreserved, and you can see clearly the cell nuclei and the cytoplasm, and importantly, there's not thick layers of ice which will impede soft X-rays. We did actually try and do cryo-sim on these samples also, but the fluorescence wasn't intense enough of our complex, and also we had an issue with the fitting of the data. So I haven't actually presented that here, but I've got a lot of tomogram conclusions that we obtained from this experiment. So again, I want to reiterate sample preparation is key. So on the left-hand side, we again have an X-ray mosaic where we've identified the cell we want to analyze. So the yellow square represents the region we analyzed by tomography. So the amount of detail you get here is incredible, at least I think so. So you can see a very well-defined nucleus, the nucleolus. You can again see the cristae in the mitochondrial matrix, dense organelles, you can see lipid droplets, and in some cases you can see the plasma membrane and features such as lamellipodia. So again, here's a short video of as you go through the tomogram, so looking at each slice, where you can see the nucleus coming into focus and different organelles appearing and coming into focus as well. Yep, so some of the conclusions we got from this. So this is particularly interesting. So this is a cell which was treated with our platinum complex under dark conditions. And what we saw was the formation of these structures which appear to look like endosomes. So this may imply that the cell is initiating the formation of endosomes in order to counteract the presence of our platinum drug, even under dark conditions. So these platinum drugs are actually non-toxic under dark conditions. So this was assumed to be because it hasn't formed the toxic platinum 2 species or released any toxic ligands. But it could also be because endosomes are contributing to the breakdown of these complexes prior to photoactivation. Another interesting observation in cells treated with platinum under dark conditions was the presence of tiny dark spots in the cell nucleus. Now, obviously with soft X-rays, they're a lot lower energy than hard X-rays, so we're not monitoring the platinum itself. We can't say where the platinum is, but we do know that heavier elements absorb X-rays more strongly, so they would appear darker in the tomograms. So we hypothesize that this might mean that platinum may be getting into the cell nucleus as a potential target. And we actually later proved this on the I-14 beamline using hard X-ray fluorescence to directly monitor the platinum. Other features we can see upon cell damage. So this is one treated with platinum and irradiated with light. We can see that a vacuole has been formed, potentially in response to the drug, so as a cellular defense mechanism. Interestingly, in this one, we can also see lipid droplets, which seem to be congregating around this vacuole. So we don't know whether the cell is synthesizing lipid droplets in response as like a repair response to repair this vacuole for cell survival. Here you can see a significantly damaged cell. We've got, you can still see the nucleus. So this is a cell treated with platinum and irradiated with blue light. Here we can see potential vesicle shedding at the plasma membrane, which can be associated with programmed cell death mechanisms. We can see multiple vacuoles. Here it's even more difficult to identify mitochondria because we could identify them very clearly before. And what I'll show you on the later slides is that this cell is significantly more flat on the grid surface as a consequence of significant cell damage. And just to end, this is a 3D segmented image of a PC3 prostate cancer cell which hasn't been treated. So here you can see the cell nucleus, you can see the elongated mitochondria in red and lipid droplets and all these features identified. And also from this, again, you can get quantitative information on the volume and size if you wish. This is the significantly more damaged cell which has been treated with platinum under photo conditions. So here you can already see that it's significantly flatter on the membrane. I know cell shrinkage can be associated with significant cell damage and also programmed cell death in many cases. Again, you can see a damaged plasma membrane and vacuoles, and also lots of dense organelles or damaged organelles in this cell. So conclusions and future work, I wanted to re-emphasize that sample preparation is probably the most important thing on having a successful experiment at B24. If you have good samples, then everything else should go really smoothly. You'd be able to acquire data very rapidly. So I think on my last experiment, we typically did 10 to 15 tomograms per day. And of course, we gain information as close to their native state as possible without having to use chemical fixatives, which may affect the cell in different ways. So again, we can gain 3D information on a subcellular level down to 30 to 40 nanometers. This is qualitative and quantitative. And we can probe drug-induced morphological changes inside cancer cells. Future work, what I think would be really exciting, at least in our field of research, is obviously at B24, cryo-sim, correlative cryo-sim and cryo-X-ray tomography is already available. But what would be really exciting is if we could then take the cells we performed tomography on to a cryo-X-ray fluorescence beamline so that we could actually monitor where our heavy metal was with respect to the fluorescence of the overall complex. So just a few acknowledgements. I'd like to acknowledge my two supervisors. So Paul Quinn at Diamond and Peter Sadler at the University of Warwick. I'd also like to thank Maria and Matt from B24 for the help with all the preliminary mapping, the experiment, and the data analysis. I'd also like to thank Carlos Sanchez-Cano for writing the proposal and being heavily involved in the experiment, and also Huayi Huang, Huayan Shi, and Ian Hans Portsman for sample preparation and any preliminary data on the complexes needed for this study. I would also like to acknowledge my sponsors, so Warwick Collaborative Postgraduate Research Scholarship, Diamond Light Source for the beam time and also the studentship, the EPSRC and Cancer Research UK. Thank you all for listening, and please let me know if you have any questions.
Thank you very much, Lizzie. That was excellent. And you've shown the process also at the beamline. We do have a few questions. I think some of them converge on the subject of your first project and to do with the mitochondria in specific. So there's been a few questions that I think in total would amount to, first, have you seen a change in the number of mitochondria you have within a treated population? And second, how do you explain the shrinkage? Do you have a hypothesis of why they shrink?
So the first question, I actually segmented 4 representative tomograms for the 4 different conditions. So we've got 2 untreated controls, dark and light, a dark treated and a radiated treated. So within these regions, so 16 by 16, we didn't see any differences in the number. And I think you'd have to analyze a bigger population in order to associate that. For example, remember, you're only taking a section of a cell, so you don't know what you're missing. It's not like the total mitochondria in the cell, so you'd have to gather more information. The second conclusions we hypothesized, so these complexes, so the iridium complexes, they can generate toxic singlet oxygen species inside cells. So the conclusions we got from this experiment were that it's likely that the damage to the mitochondria is a consequence of the generation of these singlet oxygen species in close proximity to these organelles, which is why you would see it in the irradiated samples and not the treated samples in the dark.
All right, so you mean that we're witnessing mitochondria death as opposed to mitochondria trying to respond to your drug?
Yeah, that's what I think. That's more where we're going down, like the damage to it rather than a response.
No worries. I mean, there's a related question on why do you think the lipid droplets might increase in treated cells also?
In the second?
Yes.
I mean, I guess our hypothesis was going down the lines of the cell knows there's an invader or a drug that they want to get rid of, and it's causing damage, so that they may be responding in synthesizing lipids for membrane repair or like as a cellular defense. It was interesting, what I haven't shown here is if you, in some of the cells where you've treated it with your drug and blue light, but if you recover it in complex-free media for 2 hours, you can actually see more lipids. So this is where we think that once you remove the drug, can the cell start fighting back a bit more and, yeah, release these?
And do you think that the question is, are lipid droplets electron dense? Is there a possibility that platinum is actually accumulating them?
Lipid droplets are quite dense in tomography, so I think it would be very difficult to probe whether the platinum was there, and you'll always need a complementary technique to prove that anyway. This is why we would need the cryo-sim correlative, which would be really cool, and also cryo-XRT if we have the facility.
So if you can very basically give us an idea of what generates contrast in X-ray tomography, and would you expect it? I mean, you have no experience with electron microscopy, so there's a couple of questions to compare. But just as a top level, what generates contrast in your X-ray tomograms?
So obviously at B24, you're operating at the water window. So this is very close to the absorption edges of carbon and oxygen. So in these, there are a lot of carbon dense organelles inside your cells. So particularly mitochondria, which I've shown you, but also lipid droplets tend to stand out more. But in X-ray tomography, it's not just the really carbon dense ones you necessarily see. You can also sometimes see, I think in some of ours, we've seen the endoplasmic reticulum as well, which was quite exciting. I don't know if that answers the question, but... No,
absolutely. It is a little bit more of a technical question. Would it be possible to recover a sample and maybe progress to cryo-FibSem?
You mean like take it out the beamline? I want to say yes, because I know they've done cryo-X-ray tomography. There's a paper published where they actually did then take it to a hard X-ray beamline, but I don't know specifically about that technique. But you can, if you, once you perform soft X-ray analysis, it shouldn't cause significant damage, so that you should be able to take it elsewhere, I think.
All right, and one last question. Are you depositing the X-ray data?
Depositing
in an archive?
I'm not sure actually. We've published it in one of our papers, so I try and put all the tomograms and the 3D videos I make in a mirror with the papers, that you can really see it. I'm not sure about an archive though.
Okay, so I'll just adjust this slightly because of Herald's question, and the answer is yes, we will deposit it with Empire.
Okay.
Thank you ever so much, Lizzie.